Okay, hello everyone. So I will check first of our time first. Sorry. This one, please. Okay. Before we going to further, I would like to how to say call your name about your presence in this room. Uh, Maybe this Miss Happy Kurnia Permatasari PhD. Okay.
Yes, I'm.
Yes. Mr. Michelle Alexandra.
Yes.
Oh, sorry, sorry. Oh, Miss Michelle Alexandra. Okay. So Miss Cecilia Eka Putri. Okay.
Present.
So you. And then yeah, Miss Umrotul Furgonia. Okay. Miss Umrotul. Okay.
Yes, sir.
Okay. And then Grace Ivena. Miss Grace. Okay. So Dr. Listiani Suhargo. Doctor.
Yes, I am.
Okay.
I'm asking for permission to represent Mrs. Listiani Sargo.
Okay. So Miss Indah Trilestari. Miss Indah Trilestari. Okay. Not yet come.
Yes, present.
Oh, okay. So Veronica Norman, Miss Veronica Norman.
Yes, present.
Okay, say Devi Anggrani Putri. Miss Devi Anggrani Putri. Okay, not yet come. Yeah, Miss Apriliana Noni Rahmawati.
Yes, sir.
Okay, so I would like to start our parallel section with the first presenter is Miss Happy Kurnia Permatasari from Faculty of Medicine, Universitas Brawijaya with title Effect of Essential Oil from Zigisium aromaticum, Simpogon citrate, and Melaleuca cajuput on the HeLa human cervical cancer cell viability. Time is yours, 10 minutes for your presentation.
Thank you, Mr. Rico. Hi, everyone. My name is Happy from Faculty of Medicine. So I would like to present my study that already explained by Mr. Rico. So the cervical cancer is the cervix is one of the part of woman reproduction that connecting the vagina and the uterus. So the cervix, the cervical cell, because of many factors, can become the cervical cancer. The cervical cancer from the epidemiology was around the world, and data from WHO, CDC, and other literature show that this cancer is the second most common cancer in the world, especially in women, and about 530,000 new cases by 2019, and there are about 3,700 deaths in 2019. Especially in Indonesia, there are about 40,000 new cases every year, and this is the largest amount of patients in Indonesia that usually diagnosed in the late stage. The risk factor of this cancer is environment, genetic, the lifestyle, and also the important thing is infection of human papillomavirus. This human papillomavirus is sexually transmitted virus infection that transmit into skin-to-skin contact. After this virus enter the cervical cells, this genome, the E2 genes of the virus will downregulate it. Actually, the E2 has a function as a transcriptional control for several proteins, which is the E7 and E6 oncoprotein of the virus, resulting increasing this oncoprotein. The oncoprotein E6 will inhibit or degrade the P50 3 protein tumor suppression of the host that actually regulate the G1 arrest, apoptosis, and also DNA repair. Resulting degradation of this tumor suppressor, proliferation of the cell will be increased. And the E7 will be degraded the retinoblastoma protein. This also tumor suppressor protein that related with the E2F1 cellular transcriptional factor that responsible for regulating the entering S phase in the cell cycle and also interaction with the cellular protein for cell cycle checkpoint like cyclin-dependent kinase. So the regulation of this protein will increasing the DNA synthesis and also proliferation. Both factors will induce continuity of proliferation of the cell and the cell become the cervical cancer. The current management was operative, chemotherapy, and radiotherapy, but unfortunately, operative only can be done in an early stage of the cancer. Chemotherapy and radiotherapy has a poor bioavailability and poor delivery, attacking normal cell, and therefore can cause many adverse effects. And all of these need a high cost. So then what is the solution? One is natural product that can produce the essential oil, because the previous research has shown this essential oil has a property as anti-cancer with the various mechanism of action. In here, we explore the Zizzium aromaticum that has eugenol, that has anti-neoplastic agent, and we compare with the Maleola kayu putih that has eucalyptol that has been recognized for its diverse biological and pharmacological applications such as antibacterial, antifungal, anti-inflammatory, antioxidant, and is also hypolipidemic. And also Cymbopogonardus that has citronella, that particularly known for its antioxidant, anti-inflammatory, and antimicrobial property, but these two compounds is never explored as anti-cancer. So this is graphical abstract of our study. We extract these three compounds into essential oil, and we use dose of 0.01, 0.1, and 1% of three essential oil, and then incubate with the HeLa human cervical cancers for 24 hours, and then we check the viable cell by trypan blue exclusion test under the microscope. This is the result for the Zizzium aromaticum, shows significantly decreased HeLa cell viability in a 3%, 57%, and 98%. Those increments show significant decline against the percentage of viable HeLa cell, exception only at the 0.01%, which is insignificant result compared to the negative control. And then Maleola kayu putih. After incubation with this essential oil, the viability of HeLa cell was decreased in average of 17%, 23%, and 95%. And all of three group of Maleola SP essential oil concentration show significant effect of HeLa cell viability. And then third one is Cymbopogonardus. This also can induce the HeLa cell death by the average of 7%, 31%, and 96%. And those escalation of this essential oil illustrated significant decrease of viable HeLa cells. We also compare of these three essential oil and show that this essential oil treatment strong negative correlation of essential oil administration toward the cervical cancer cell viability as shown by Pearson correlation coefficients. And for the concentration of 0.01, this is the lowest concentration that we use. The Maleola kayu putih essential oils show more robust reduction of the HeLa cell viability compared to the other two. And for the highest concentration, so all of the three essential oil exhibit the similar potential cytotoxic activity. And we also determine the IC50 for the Zizzium aromaticum. It is 64.16 microgram per milli, and also Maleola kayu putih about the IC50 is 111 microgram per milli, and for Cymbopogonardus it is 95.12 microgram. So according with the IC50 value, cesium aromaticum essential oil was most potent to induce cell death as it exhibit the lowest IC50 among the others. Based on the IC50, we check the apoptosis of the cell using three doses, 30 microgram per milli, 60 microgram per milli, and 120 microgram per milli, and incubate with the HeLa cell. And we use the annexin 5 and PI, and then for further analysis, we use flow cytometry to check the positivity of annexin 5 or PI. So for the cesium aromaticum, this one also induced apoptosis of HeLa cell in the dose of 60 microgram per milli. It can induce almost 80% of the cell HeLa. For Malacca kayu putih, to induce 80% of the apoptosis of the cells, it is need a higher concentration. It is about 120 microgram per milli. And then for the Simbobagun nardus, it is also familiar with the kayu putih. The effective dose to induce almost 80% of apoptosis was 120 microgram per milli. So conclusion of our study, one, essential oil treatment significantly reduce HeLa cell viability in a dose-dependent manner. And in low dose of treatment, Malacca kayu putih essential oil show more robust HeLa cell viability reduction compared to the other plant essential oil. And cesium aromaticum was most potent to induce cell death. If we see the IC50, it was 64.16 microgram per milli. And for cesium aromaticum essential oil, also most potent to induce apoptosis in the 60 microgram per milli compared to other two essential oil. It is about 120 microgram per milli to induce apoptosis more than 80% of the HeLa cells. Thank you.
Thank you very much for your presentation, Dr. Happy. So for the how to say it, like question and answer, we will going to the question and answer question. So for the next presenter is Miss Michelle Alexandra Kanandi from Vidya Mandala Catholic University Surabaya with the title The Effect of Extraction Method on the Yield and Antiplatelet Activity of Compounds in Gentiana Plant Extract, a literature review. Miss Michelle Alexandra, time is yours.
Yes. Okay, thank you, Mr. Rico. Good afternoon. My name is Michelle Alexandra Chanandi. Today I'll present about my research about the effect of extraction method on the yield and antiplatelet activity of compounds in Gentiana plant extract, a literature review. So uncontrolled platelet response can lead to thrombus formation, which could lead to ischemia or infarction. This could be treated with antiplatelet drugs, but even though antiplatelet drugs are widely used, the risk of hemorrhage and antiplatelet drug resistance push the development of new drug to control thrombus formation. One of the potential to be used as new drug development are plants such as the Gentiana plants. The Gentiana plants are have many effects such as anti-inflammatory, analgesic, diuretic, hypoglycemic, and have shown antiplatelet effects in the cardiovascular system. As it is a natural product, there is a need for effective extraction method to gain high yield for further research and development. So for the literature review process was done from November 2020 until February 2021 on 4 database, Science Direct, PubMed, GoScholar, and ProQuest. For the keywords used are the Gentiana species, compounds containing Gentiana extract, antiplatelet activity, and extraction methods. For the inclusion criteria are all articles within the last 20 years that is related to the topic of the research, and the exclusion criteria are all articles that cannot be fully accessed by the authors. And as you can see from the scheme, from 150 literature that we found, it was screened, and in the end, 20 literature was used in the literature review. So based on the literature, there are 7 compounds in the gentiana plant that has an antiplatelet effect. That is orientin, amorgentin, vitexin, isovitexin, mangiferin, and oleanic acid. These 4 are flavones. This is a xanthan, and this is a coiridoid glycoside, and this is a triterpenoid. And for the antiplatelet activity, oleanic acid show 2 contradictory activity that it could enhance and inhibit the platelet aggregation. And based on what we summarized, it was because of the different dose used in the research, that at a low dose, it acts as a stimulant for platelet aggregation, and at high dose, it acts as an inhibitor for platelet aggregation. For mangiferin, it has an antiplatelet mechanism similar to its derivative, noratriol, by inhibiting the arachidonic acid and EDP pathways, which will in turn inhibit the glycoprotein 2B3A activation. For amorogentin, it inhibits the collagen-induced platelet aggregation by inhibiting the MAPK pathways and the phospholipase C gamma 2 enzyme. For the orientin, vitexin, and isorientin, they also inhibit platelet aggregation through various pathways such as collagen, thromboxane A2, and EDP receptor, which will then inhibit the activation of glycoprotein 2B3A. From all the literatures, it was summarized that the compound with strong antiplatelet effects were amorogentin, vitexin, and oleanolic acid. The compound with the strongest antiplatelet effect was amorogentin, with the smallest IC50 of 30 micromolar. And then for the factors affecting the extraction yield of gentiana extract, there are 4 factors. That is the different method of extraction, the duration of extraction temperature, type of solvent, and concentration of the solvent. Various extraction methods were used in the journals. Some use cold methods such as maceration and ultrasonic, while other methods such as chocolate, heating, infusion, and digestion. The extraction process was done and repeat 1 until 3 times, and the duration was from 10 minutes until 2 days. The solvent that is used in the literature were water, ethanol, and methanol. For the duration of extraction, the longer the duration of extraction, the higher the concentration of the yield obtained up to a certain extent. And the extraction time of a hot method is usually quite short compared to the cold method due to the faster extraction speed and to prevent the heat degradation of the compounds. The use of other aids such as ultrasonic waves can shorten the extraction time. For the extraction temperature, the use of high temperature will increase the solubility and rate of diffusion so that a high yield of compounds can be obtained. But we also need to see the compound stability, because if the compound is unstable to heat, degradation might occur and result in lower yield of compounds. Some of the unstable compound in heat is like mangiferin and amorogentin. For the type and concentration of the solvent, in the dissolution process, a compound will be easily dissolved in a solvent with similar characteristic as the compound. From the literature, it can be concluded that the extraction of fungiferin, vitexin, orientin, isorientin, and isovitexin is recommended to use water or low concentration of alcohol as solvent. And for amorogentin, it is more optimally extract using a 60 to 80% concentration of alcohol. And for oleanilic acid, it is recommended to be extracted using a nonpolar solvent. Okay, so for the conclusion, the extraction method, duration of extraction temperature, type and concentration of solvent will determine the content of compound in the Gentiana extract. And the second conclusion, each compound has antibiotic activity with different mechanism, and the strongest one is amorogentin that acts by inhibiting the collagen pathway IPA. That's it about my presentation. Thank you for your attention.
Thank you very much for Miss Michelle. So I'm going on to next presenter is Cecilia Eka Putri from Atmajaya Catholic University of Indonesia with the title Effect of Chili Extracts on the Modeling Energy Homeostasis Related Gene in Obese Mice. For Miss Cecilia Eka Putri, 10-minute time is yours. Miss Cecilia.
Thank you.
Okay.
Good afternoon, ladies and gentlemen. My name is Cecilia. Today, I would like to present our research about effect of chili extract on modulating energy homeostasis-related genes in obese mice. Chili is widely used as flavoring spices in many dishes. The major active component of chili is the pungent capsaicinoid, which consists of capsaicin and dihydrocapsaicin, which give us the hot sensation. And the other component is capsinoid, the non-pungent component from chili, antioxidant, vitamins, and carotenoid. Chili contain capsaicin, which have anti-obesity mechanism, which partially similar with orlistat, the commercial anti-obesity drug. However, orlistat has side effect in long-term usage. Capsaicin is the main active component in capsicum fruit. The characteristic of capsaicin is an alkaloid compound which have odorless, white crystal, and water insoluble. The capsaicin content in chili virus depends on the type of chili. The level of capsaicin range between 1 microgram per gram in green pepper up to 4,000 microgram per gram in hot chili. Capsaicin can suppress hunger, induce satiety, enhance lipid metabolism, enhance thermogenesis, inhibit pancreatic lipase and alpha amylase activity, and prevent adipocyte differentiation. This is the structure of capsaicin. From the structure, we can see that capsaicin has the similar structure with the pentermine and orlistat. The anti-obesity mechanism of capsaicin is to increase the energy expenditure and lowering the food intake and inhibit the lipase activity. Leptin and obesity. Leptin is a hormone that produced by adipose tissue. Leptin regulates the food intake and satiety. Leptin will send signal to hypothalamus. When level of circulating leptin is high in the blood, it will send the signal to hypothalamus that our body is full. And the hypothalamus will send the signal to lowering the food intake. Obesity is the condition when our body is in high body fat. The higher body fat will produce more leptin, but not the leptin receptor. And if it uncontrolled, it will cause the leptin resistance. This is the picture of signaling mechanism of energy homeostasis in leptin melanocortin pathway. Leptin, which produced in adipose tissue, will bind with the LAPR, the receptor, and the binding of leptin and its receptor will upregulate the the production of PUI-PT. And in the end, it will increase the energy expenditure and lowering food intake. The objective of our research is to test the effect of various chili extract on modulating energy homeostasis related genes. There are LAPR, SIM1, PUMC, and HH2B1 in obese mice. Materials and method. This is the chili that we use in our research. There are raw chili, green chili, and red chili. The method, the research is conducted by following this kind of method. First, we extract the dry chili to collect the chili extract. After that, we do in vivo test on mice which treat with standard and high-fat diet. The parameter obesity, such as total cholesterol, triglyceride, and body weight, were measured within 30 days. After that, we do RNA isolation using RiboX. And then we do qPCR analysis with kappa master mix. The expression of leptin-related gene, there are LAPR, SIM1, PUMC, and SH2B1 were measured against the housekeeping gene GAPDH. After that, the expression of gene were analyzed using SPSS, one-way ANOVA to KISS post-hoc. This is the primer that we use in our research. And this is the PCR condition that we use in our research. Result and discussion. This is the total capsaicin content from chili extract. From this table, we can see that raw chili extract contain about 45 milligram per gram of capsaicin, and green chili and red chili have the similar amount of capsaicin. There are 52 microgram per gram extract. And from this graphic, we can see the total cholesterol and triglyceride from mice treated with chili extract from which observed in 2 and 4 weeks. From this graphic, we can know that giving the chili extract can reduce the total cholesterol and triglyceride from obese mice. And from this figure, this is the figure of LAPR gene expression after treated with various chili extract in adipose tissue and hypothalamus. From this figure, we can see that CR25 treatment had the highest LAPR gene expression value, which are 10.3 times in adipose tissue. And also in hypothalamus, CR25 treatment show a significant upregulation of LAPR gene compared to control positive. The previous study said that obesity caused elevation in circulating leptin, but had deficiencies of leptin receptor. Upregulating the LAPR will increase the ability of leptin receptor to bind the leptin. Next, this is the figure of SIM1 gene expression after treated with various chili extract in adipose tissue and hypothalamus. From this figure, we can see that all chili extract from raw chili and green chili can upregulate the expression of SIM1 gene. And CR25 treatment has the highest SIM1 gene expression in adipose tissue and hypothalamus. The previous study said that insufficiency of SIM1 in mice cause obesity by increasing the food intake. High expression of SIM1 will improve the signaling of MC4R, which regulates in decreasing food intake and increasing energy expenditure. Next, this is the figure of POMC gene expression in adipose and hypothalamus tissue. From this figure, we can see that chili rawit extract at concentration 25 milligram per kilogram can upregulate the expression of POMC gene in adipose tissue and hypothalamus. The previous study said that expression of POMC was sufficient to maintain normal food intake. POMC will stimulate alpha and beta MSH, which contribute to suppress food intake stimulate energy expenditure, and enhance insulin sensitivity. Similar to our result, the ability of chili extract on upregulating POMC gene expression may contribute on prevention of obesity by suppressing food intake and increasing energy expenditure. Chili extract also has ability as anti-obesity agent by enhancing the insulin sensitivity. Next, this is the figure of SH2B1 gene expression after treated with various chili extract in adipose tissue and hypothalamus. From this figure, we can see that chili rawit extract at concentration of 25 milligram per kilogram can upregulate the expression of SH2B1 gene in adipose tissue. SH2B1 activates the JAK2 cytoplasmic tyrosine kinase to mediate cell signaling. SH2B1 was known as leptin sensitivity regulator. Upregulation of SH2B1 will increase the leptin receptor ability to control energy balance and body weight through enhancing leptin signaling. And in the conclusion, from this research, we can conclude that capsicum can be extracted to a chili extract, which rich in capsaicin. The chili extract then used in vivo test on mice, and the obesity parameter was observed for 30 days. After that, the mice were euthanized and the organs were collected, and we isolate the RNA using RiboX. After that, we run qPCR for those genes. There are LabR, SIM1, POMC, and SH2B1. And from this research, we find that giving chili extract from rawit chili at 25 milligram per kilogram body weight has a promising anti-obesity agent that has functional effect to modulate energy homeostasis related gene in obese mice. I think that's all. Thank you for your kind attention.
Okay, thank you very much for Ms. Cecilia Eka Putri. So we are going to next presenter is Ms. Umrotul Furgonia from Universitas Erlangga with the title Synthesis of Exploration and Exploration of dihydropyrimidintione derivative activity as human neutrophil elastase HNE inhibitors. For Ms. Umrotul Furgonia, you have a 10 minutes for your presentations. For your presentation, time is yours.
Hello everyone. Allow me to introduce myself. My name is Umratul Furgonia, and my counselor are Mr. Heri Sito and Mr. Riko Ramadan. Now I will present about research with title Synthesis and Exploration of dihydropyrimidintione derivatives activity as human neutrophil elastase inhibitor. Before going further, we need to understand what the human neutrophil elastase enzyme is. Human neutrophil elastase, or HNA, is an enzyme that has activity against infection and as the body's defense against foreign protein. HNA activity regulated by endogenous inhibitor or antiproteins such as ellatin. But how if actual concentration excess of HNA? We know that anything excess is not good, right? Similarly, if the amount of HNA enzyme in the body is excessive, there will be a protease and antiprotease imbalance. So it can cause tissue damage and lung function disorder. One of them is chronic obstructive pulmonary disease or COPD. It can be relieved by the addition of an exogenous HNA inhibitor to build the protease and antiprotease balance. Has there been any research about the inhibitor before? Yes. Previous HNA inhibitor that have been discovered as shown in the figure. However, based on study of 1,500 COPD patients showed that there were no significant clinical outcome for these drugs. In 2015, research by von Baum showed that 1.4 dihydropyrimidinone derivatives had good potency as HNA inhibitor. Besides being able to inhibit HNA, candidate molecule must also have antioxidant activity. Unfortunately, the amide group contained in the structure of 1.4 dihydropyrimidinone compound has weaker antioxidant activity than the thiamide group. Therefore, in this study have been synthesized another compound that is dihydropyrimidintione, which is expected to have better activity. A retrosynthesis analysis as shown on the left figure. The retrosynthesis shows that there are two steps of synthesis. They are urea synthesis and dihydropyrimidintione synthesis. In this research, synthesis of dihydropyrimidinedione based on the synthesis method by Hadiansyah in 2019, but this synthesis takes a long time, which is 22 hours. Therefore, in this research, we made modification to the Hadiansyah method that is synthesis without solvent. Based on the background that has been described, we report and discuss the effect of solvent, substituent variation of phenylethylurea and benzaldehyde reagent in the synthesis of dihydropyrimidinedione derivatives, also their potency as a chain inhibitor based on the in silico studies. To find out the method used, let's move to the experimental section. The following are the materials, instrument, and software used in the research as shown in table. After all of the material are prepared, the first step is mix 3 reagent, also paratoluene sulfonic acid as a catalyst. The second step is heating the reagent mixture accompanied by stirring. The reaction is complete. The reaction mixture is cooled and extracted using suitable organic solvent and distilled water, followed by the precipitation of the product using a suitable solvent. Then the product obtained is characterized. Activity exploration of the target molecules through the in silico consists of two stages. They are molecular docking and molecular docking. The receptor used for the in silico studies was derived from the PUI-PT complex 5A0A. The steps in the molecular docking stage are preparation of ligands, receptor, surface and sphere, also box and butyration, then docking process. The dock ligand is followed by a molecular footprint type to determine the interaction between the ligand and amino acid residues of receptor. Then the data obtained are energies of van der Waals interaction, electrostatic, and the presence of hydrogen bonds. The results of this research are follows. Based on its structure, the target molecules can be synthesized from the benzaldehyde, phenylethylurea, and acetal compound through the binary reaction. In immuning mechanism, continuity of the binary reaction determined by nucleophilicity of phenylethylurea and electrophilicity of benzaldehyde, which is influenced by substituent attached to the aromatic rings of each component. The basic structure of the target molecules contain two aromatic rings bound to the of the number 1 and carbon number 4 atoms, which assumed to interact with the S1 and S2 active sites of HME. Substituent variation bound to the two aromatic rings also affect the interaction that will form between the ligand and the receptor. Besides synthesis all of target molecules, solvent optimization is carried out first. Solvent optimization is carried out in the synthesis of molecule target 1 using several kinds of solvent and solventless. Optimization results showed that the synthesis of molecule target 1 with solvent condition produced the highest yields and the fastest reaction time. So these conditions are used in the synthesis of other target molecules with a variety of substituent on the reactant component of phenylethylurea and benzaldehyde. The reaction time and yield data obtained from the synthesis are shown in the table. The synthesized product was tested for purity using TLC with two different ion systems as shown in the table. After being declared pure, then proceed to the characterization process. The first characterization is high-resolution electrospray ionization mass spectrometry, or HRICMS. Characterization using HRICMS was used to determine the molecular formula and relative molecular mass of the target molecules as data shown in the table. In general, the analysis results show a peak of positive ion molecules, M plus H plus. Based on the results, it's also known that molecule target 1 to 8 have the same degree of unsaturation, which is 12. The next is FTIR characterization result. Based on FTIR spectra, on the structure of molecule target 1 to 8, there are secondary N single bond H, C single bond HSP2, C single bond HSP3, C double bond O, C double bond C, C double bond S, also C single bond C. In addition, there are other absorption bands that are only present in the spectra of certain target molecules as shown in the table. And the following is the wave number data of each target molecules. Based on proton NMR spectra of the target molecules, in general there are 5 types of proton signal. They are proton of nitrogen number 3, carbon number 4, carbon number 7, carbon number 9, also aromatic proton. Proton signal of nitrogen number 3 and carbon number 4 appear as doublet with the same coupling constant value. Then proton signal of carbon number 7 and carbon number 9 have singlet multiplicity and integration equal to 3. While the aromatic proton signal have various multiplicity, it influenced by difference in the substituent. The following is a table of the proton signal chemical shift in the core structure of the target molecules. NMR carbon spectra of target molecules 1 to 8 show the number of carbon signal according to the structure of each target molecules. In this spectra, there are 2 quaternary carbon signal. They are the most disorbing. They are carbon octet carbonyl and carbonyl group. There are differences in the carbon signal in each target molecule spectra due to different substituents as shown in the table. The UV spectra showed that almost all of the dihydropyrimidinedione derivatives had 2 absorption peaks. Based on the structure of the target molecules, there are 3 kinds of electron transition that can occur. They are non-bonding to p star orbital at 221 nanometer region and p to p star at 306 nanometer region. After we know the result of the target molecule synthesis and their characterization, then what is the effect of different substituent of the reagent? The substituent of phenylurea affects electron density around the nitrogen atom of primary thioamide group. The electron donating substituent in phenylurea can increase electron density around the nitrogen atom of primary thioamide group, thereby causing the nucleophilicity of the nitrogen atom of the primary thioamide to increase. While electron withdrawing substituent will decrease the electron density in the aromatic system and the carbonyl group. As a result, the electrophilicity of the carbon atom of the carbonyl group increases and is more susceptible to attack by the nucleophile. The next is molecular docking results of target molecules. The in silico analysis of activity potency of the target molecules consists of two stages. The first step is docking of target molecules with R and S conformation. Based on the molecular docking result, the target molecule with the best docking score and inhibition constant is RMP1, with our molecule target 1 with R conformation. The docking score and inhibition constant of RMP1 almost the same as reference ligand. These results show that the stability of the interaction formed between RMP1 and HNA is almost same as the stability of interaction between the reference ligand and HNA. It can be seen in the figure that RMP1 is bound at the same position as the reference ligand, and the two aromatic rings of the target molecules enter and interact with the S1 and S2 active sites of HNA. The second stage of in silico test is molecular footprint. Based on the molecular footprint studies, the reference ligand footprint pattern was obtained, which showed that the amino acid residues that contribute the most to form van der Waals interaction with the reference ligand and RMP1 were histidine 41, tyrosine 80, leucine 85, aspartic acid 88, phenylalanine 168, serine 171, and phenylalanine 187 residues. Then are these interaction as shown in the table. The negative value of the interaction energy show attraction interaction, while positive value show repulsion interaction. Electrostatic interaction also occur between amino acid residue in HNA with reference ligand and RMP1. The residues that most play a role in forming electrostatic interaction with the two ligands were histidine 41, tyrosine 80, aspartic acid 81, leucine 85, leucine 86, aspartic acid 88, arginine 151, serine 186, and valine 188, with the interaction energies as shown in the table. Footprint pattern of the van der Waals and electrostatic interaction showed many similar interactions between the amino acid residues of HNA with the reference ligand and RMP1. While the interaction dissimilarity was found in several amino acid residues in the same region that are residues 80 to 86. The difference in interaction was caused by the difference in the substituent attached to the aromatic ring of both compounds. In addition to the van der Waals and electrostatic interaction, there are one hydrogen bond between HNA and the ligand, both the difference ligand and RMP1 ligand. A hydrogen bond is formed between the NH of amide group on the valine 188 residue with oxygen atom of carbonyl group in the reference ligand, also sulfur atom of thiol carbonyl group on the RMP1 ligand. Based on the result of docking and molecular footprint, the interaction formed between the ligand and HNA is visualized in the figure. The reference ligand and RMP1 form four kinds of interaction with the amino acid residue of HNA. They are van der Waals, electrostatic, hydrogen bond, and PP stacking. Van der Waals interaction on both ligands occur between two aromatic rings bound to the nitrogen number 1 and carbon number 4 atoms with amino acid residue on the active sites of HNA. The aromatic ring attached to the carbon number 4 atom and the hydropyrimidinol ring of the reference ligand form PP stacking interaction with the histidine 41 residue. While the PP stacking interaction of RMP1 ligand occurred between the hydropyrimidinol ring with histidine 41 residue and the aromatic ring bound to nitrogen number 1 atom with phenylalanine 168 residue. The conclusion of this research are follows. The first is synthesis of target molecules derived from the hydropyrimidinol through the Divinelli reaction without solvent increase the yield of the synthesis product and speed up the reaction time. Second, substituents of benzaldehyde compound affect the electrophilicity of the carbon atom of the carbonyl group. Meanwhile, substituent of phenylurea compound indirectly affect the nucleophilicity of nitrogen atom of the primary thioamide group. The greater the nucleophilicity of phenylurea and the electrophilicity of benzaldehyde, the easier progress of reaction and affects the yield obtained. The third, based on the in silico studies consists of molecular docking and molecular footprint, RMP1 compound has potency as HNA inhibitor almost the same as the reference ligand. Okay, that's all of my presentation. Thank you.
Okay. Thank you very much for your presentation, Ms. Umratul Furgania. So we're going to the question and answer section for 10 minutes. So now from chat box, I already have for question from the others participants. The first question from Dr. Armando from UNER. He asked for Dr. Happy that um, talking about like when you said that the solution treating the cancer just the treatment strategy does this attack normal cell, so how you can make sure that your plant extract did not affect for normal cell? Maybe for address first, Dr. Happy.
Okay, thank you so much for the question. So yeah, actually the normal cell and the cancer cell has a different gene expression and also protein expression. So we hope that our plant extract, the target is the protein that express only the cancer cell, but not the normal cells. So this is only the initial study. We will continue our study. We will use the normal cell like Higgs 29 or other cell, and then compare the effective of this plant toward the normal and the cancer cell. For example, previously we already explored about the eugenol that from the Zyzium aromaticum. This is for the cancer cell. The dose is lower than the normal cell. So in the effective dose for induced apoptosis of the cancer cell is lower compared to the normal. So the normal will be toxic if we use the highest dose. We hope for the other two plants. Of course, this is not already done now, but we will study in the next study.
Okay, I think for the second question also like the same that your
Almost,
explanation, right?
yes.
Yes.
Yeah, but
So
they said about the side effect. For the side effect, we will not use the in vitro study, but the in vivo study, because the side effect is the effect that we don't want to. And then effect for the other cells or the other tissue. So we will use the in vitro study, and then we will explore and analyze the effect to the others organ for the effect of this plant. So but we use the natural plant that already used widely. So we hope this is safe for the normal cells.
yeah, I see. So thank you very much for your address the question, Dr. Happy. So for
You're
next
welcome.
one, yeah, the next one is the question for Miss Michelle. Miss Danang from Universitas Erlangga will ask you about um, what the effect type of solvent to get a compound gentian plant extract. Maybe you can short explain what the effect type of solvent to get a compound in the gentian plant extract.
Yes.
Yes,
If
go
you
ahead.
use polar solvents such as water or low concentration of alcohol, there will be more polar compound extracted in the extract. And if you use non-polar solvent, there will be more non-polar compound that is extracted.
Okay. So that is you mean like using the solvent based on the compound that you want to extract, right?
Yes.
But yeah, okay, I see. So thank you very much for your address question, Miss Michelle. So for next question is come from Rizky from Universitas Erlangga. Um, he is curious about the from research of Miss Cecilia. That the question is based on your research, right? That primer you have that is from literature or you self-design, and then that is need optimization. Maybe you can address Miss Cecily about this question.
Thank you, Mr. Rico.
Yes.
For Rizky, thank you for the question. The primer that we use in our research is we design all the primer by ourselves using primer tree software, and the sequence of the mice, whole genome sequence of mice is from the NCBI. And if it is need for optimization, yes, it's need we do optimization by optimize the optimal annealing temperature. Thank you.
Okay, thank you very much for your just question. So we're going to a next session still in the oral presentation. So